Microtubule dynamics have a significant physiological effect on the cell. Sometimes cells have to make polymer very, very rapidly. Sometimes it has to depolymerize. If cell was dependent upon a strategy that every time it had to make polymer, it has to first of all synthesize the protein. The proteins have, as we know, it would take time. Messenger RNA will be produced. It will result, the messenger RNA will interact with ribosomes. Those ribosomes will ultimately form the protein. The mature proteins, as we know, some of them, they go through different modifications also. So if cells were to follow this strategy, it would take them much longer to respond to a particular change in the environment or respond to a particular signal. A better way to do this is cells can overproduce the overproduce the monomer, sequester the monomer, so it is out of the polymerization pool, and only release the monomer when and where cell needs to polymerize more cytoskeletal elements, actin, and also microtubules. We saw how that strategy worked in case of actin. Now, let me show you how this works in case of microtubules. Stethmen is a protein that can sequester monomers of tubulin, the alpha and beta dimers. Stethmen does that. Let's talk about that. The unpolymerized tubulin subunits are sequestered in the cell to maintain the subunit pool at level substantially higher than critical concentration. And this job is done, as I mentioned, by Stethmen. One molecule of this protein, Stethmen, binds to two tubulin heterodimers and prevents their addition onto the microtubule ends. Stethmen decreases the effective concentration of tubulin subunits that are available for polymerization. This action is basically analogous to that of colchicine that we have talked about. High levels of active Stethmen in cell decrease the elongation rate of microtubules. The transition from growing state to the shrinking state for a microtubule, GTP hydrolysis, and filament elongation, slowing the elongation rate by sequestering the subunits, can increase the frequency of microtubule shrinkage, which makes sense. So if you have, let me show you a diagram of this. So if you have Stethmen, here's the blue molecule. This blue structure is the Stethmen. This Stethmen is, as I mentioned, one Stethmen molecule can bind two of these tubulin dimers. So the light green and dark green balls, they're the dimer, tubulin dimer, and one Stethmen molecule is binding two of these. So when the Stethmen binds these free tubulins, molecules, subunits, it sequesters them, and these subunits cannot then add on to the microtubule. So effective pool of microtubule subunits is reduced. When that happens, if no microtubule subunits are being added, the microtubules can lose the GTP cap, and once that happens, microtubule, as we have said, that beta tubulin bound to GDP by default forms these curls. It's no longer straight, and when the structure curls up, these protofilaments curl, the microtubule basically starts to depolymerize. So a protein that inhibits tubulin polymerization can have a secondary effect by dramatically increasing the dynamic turnover rate of microtubules in the cell. So cells use similar strategies for controlling microtubule polymers and also actin polymers, which is basically sequestering the monomers and reducing the effective monomer population. This gives cells the flexibility to quickly polymerize the filaments, the microtubules or actin filaments, when and where they are needed.